Okay, so again, chapter 6, metabolism, the introduction to how we make energy. This will dovetail into the very end, looking at photosynthesis and cellular respiration, and how ultimately those next two chapters, how plants and animals, us, make the goods, make all that energy that we need to do work. So what is energy? Well, crap, I just said it. Ultimately it's the ability to do work and bring about change. But I mean, for all intents and purposes, this is what it is. It's the ability to do stuff, okay? Bring about change in a cell, bring about change within a system, circulatory system, the biosphere, whatever system, whatever level you're talking about, energy is what is ultimately needed to do the work. We have two types of energy. You've probably seen them before. Kinetic energy, which is energy of motion, okay? Energy of motion. And potential energy has the potential to do stuff, and it is stored, okay? It is stored energy. An example of kinetic is mechanical, okay? Picking up stuff, moving it, okay? I'm gonna pick up this thing and move it, okay? So that is kinetic energy. It's energy of doing stuff. Water going over a waterfall is the classic example of that, all right? An example of kinetic would be mechanical energy. You picking up a rock and throwing it, all right? You picking up the baby, all right, and walking around, energy of motion. Potential is that stuff that we have in food, all right? The chemical energy that it's found within the food, the carbohydrates, the lipids, the proteins, okay? Those macromolecules, those biomolecules, all right? It's the food that we eat. That food that we eat has chemical energy stored in it. And then it's our digestive system's process to break it down and harness all of that potential energy so we can utilize it inside ultimately the mitochondria to make ATP, adenosine triphosphate. All of this energy starts at one spot. The sun. Energy starts with the sun. Plants utilize that sunlight. They are autotrophs. They make their own food, right? They're photosynthetic. So they harness that solar energy and make chemical energy. What in the hell do they make? Carbohydrates. They make carbohydrates and they'll utilize it for their structure to do stuff. Or they store it, and then heterotrophs, okay? The moose, it's going to utilize that carbohydrate inside the plant, and it's going to utilize it to make energy so it can do work. Walk around in the field. And all the while, as this energy is flowing, as you can see from the picture there, we give off heat, okay? Or heat is given off, I guess I should say, all right? When energy goes from one form to the next, from solar energy to the chemical energy, the processes that are going to be occurring in photosynthesis that we'll talk about on Wednesday, we get this change of energy, okay? And as we go from one form to another, we give off energy, we give off heat, okay? Because no transition, no making of energy from one form into another is ever 100%. So we give off this thing called heat. It's a waste product. When we talk about energy, we ultimately have, or the flow of energy, I guess I should say, we have two basic laws, okay? They're called the laws of thermodynamics. These laws help us to explain how energy is moving through a system. Being able to potentially discuss this on an exam would be a good potential short answer question, okay? And we have two laws. You should definitely know what the first law and second law is, and then you definitely should be able to explain it, okay? Because you know it's going to be on the exam. You know it has the potential to be a good multiple choice question, but it has a damn good chance of being a good, fabulous short answer question, essay question, whatever you want to call it. So we have two laws. The first law is the law of conservation of energy. Okay, and this is basically what it says right here by that star. Energy cannot be created or destroyed. It is merely changed from one form to another. So going from, like I said in that previous slide, plants, capturing that photosynthetic energy, okay? Capturing that solar energy from the sun. It takes it and helps to transform it into carbohydrates, okay? Gives off another waste product, oxygen, which we will then utilize and then will help to fund our ability to make energy. And then our waste product is CO2, and that's what plants utilize, okay? So they're going to take sunlight, they're going to take carbon dioxide, and they're going to take water, and they're going to mash it all together and help to form carbs, carbohydrates. The transformation of that solar energy plus carbon dioxide plus water, okay, ultimately forms carbs, but it also gives us waste products of heat and ultimately oxygen. The second law is the law of entropy, and entropy is just disorder, okay? It's just disorder. That's all it is, okay? It states basically right here, this is the definition for it there for each star, okay? When energy is changed from one form to another, there is a loss, okay? Nothing is ever 100% efficient, okay? There is a loss of energy. That loss of energy is in the form of heat, okay? Especially when we look at humans, because that's what I obviously focus on a lot more than plants. But look at how we make energy with cellular respiration, okay? We'll talk about photosynthesis Wednesday, and then next week we'll talk about cellular respiration, how we make energy, okay? We are only like 33%, 36% efficient, depending on the cell. That ain't no near 100%, is it? Okay. So we are not very efficient in making this a thing called energy. And that's in the presence of oxygen. If you're in a presence of not enough oxygen, it's only 2% efficient. Good God Almighty, that's not even nearly 100%. Okay. So no process in the formation of energy, getting it, harnessing it from the potential of within the carbohydrate or within the fat or within the protein that you eat. All right, as we harness that energy or extract that energy from these molecules, process is not 100%, not even close. The vast majority of it is always lost as heat. And then the body has multiple different ways to help get rid of that. And we talk about some of those within, human anatomy or physiology and stuff like that. So here we go. I mean, ultimately, here it is. Okay. Energy is not, they don't create it from nothing. They take it from the sun, take that energy, that solar energy, and change it over, make a carbohydrate. So they've made something from something else. First law. Second law. Nothing's 100% efficient. Okay. And heat is that byproduct. We could take that carbohydrate and do stuff. We could take that carbohydrate and utilize it to make ATP. To move. And in the process, give off heat. Okay. So when we start looking at photosynthesis and cellular respiration in the next week, you're going to see that they're inherently connected to one another, right? Because again, it all starts with the sun. Here is entropy, and entropy is just disorder, okay? Disorder. Every time I say that, I think of that. That song. Toxicity. Okay, sorry. break for it. So here you can see glucose, right? Carbohydrate. Very, very organized. Look at that thing. Oh my God, that has a beautiful structure to it. And all of those bonds that are holding this thing in its nice organizational structure, all of those bonds, they have so much potential energy within them, okay? You can see when we send it through the digestive system and we break it up, now it's not organized at all. Water and carbon dioxide has really nothing in it. It's just waste, okay? We've harnessed all the energy out of the good bonds, and this is the leftovers, okay? It's more stable because there's a lot of disorder, okay? It's just bouncing around, it's moving around, okay? Look at the bottom there. On the bottom picture, we have a plasma membrane, right? With an integral protein, one of those channel proteins. We have an unequal distribution on this side, okay? Body doesn't like that. It's going to try to get some of these across because it wants to be balanced on both sides of that plasma membrane. So it's going to send some through. And in the process, we're going to harness some energy out of that, pumping these hydrogen ions across, ultimately. Okay, and we'll see that Wednesday. We'll see that Monday next week when we look at photosynthesis and when we look at cellular respiration. Okay. Okay. Every process in a cell increases the total entropy, this total amount of disorder. Okay. Therefore, less energy to be available to do work. Think of a nice clean room, organized, a lot of potential for it to be messed up, okay? That's basically glucose. You go in your room, messy bed, clothes everywhere, there's a lot of disorder, okay? And now there's not a lot of potential to make it any messier because it's already a pigsty, right? That's basically how you should think about this concept, okay? Once there's disorder, we've basically harnessed all the energy out of it. So there's no potential in it anymore, okay? It's already messed up. Here's some words to that, what I was talking about at the very beginning with that picture, same picture as you can see there. These are my producers. Okay. They're autotrophs. Okay. A-U-T-O-T-R-O-P-H-S. S. They make their own food. Make own. That's a W. Food. Okay. Again, they make their food. They make that nice organized structure, carbohydrate, right? Okay. And then these guys here, these heterotrophs, H-E-T-E-R-O-T-R-O-P-H-S, They can't make their own energy, okay? So they're going to go and eat the producer, the plant. So we need a constant flow of energy. We need a constant supply of solar energy for this entire system to function, okay? Again, have a lot of potential within that glucose, within that carbohydrate. The moose, the heterotroph, eats that carbohydrate. And then through the digestive system, breaks it down into those monosaccharides. Breaks down glucose, breaks down those monosaccharides, okay, to try to get all the potential energy out of those bonds. And ultimately form waste, water, CO2. Both of them are picked up by the bloodstream and gotten rid of, exhaled, urinated out, whatever the case may be. But all of this system is dependent upon one thing, Mr. Sun, okay? Mr. Sun. If we don't have Mr. Sun, none of this system works, okay? And the system falls apart. What if we don't have enough sun for the autotroph? We have different types of photosynthesis. Sometimes plants, the conditions aren't conducive to make carbohydrates and to make energy for itself to do stuff. So we have different plants and we have different environments, and so we're going to have various different types of photosynthesis to ensure that we can fully harness Mr. Sun. So we can make ATP, or the plant can make ATP and make carbohydrates and do stuff, which ensures that we can have stuff to do stuff as well. Okay, because even though I may not be out grazing on a plant, I'm going to eat a little bit of plants, not get much out of it, fiber, a little bit of carbs, okay? I don't have a lot of enzymes to digest it, but ruminants do. A lot of animals do. And I'm going to eat the animal. Okay? I'm going to eat the animal. And I'm going to get that energy. I'm going to get all that protein. Okay? Carbs. Fats from my diet to fund me. So when we talk about all of these chemical reactions that are happening within a cell, we are talking about metabolism. And that's ultimately what we're talking about today, right? Metabolism, by definition, is just the sum of all the chemical reactions in a cell. Okay? All the photosynthetic things that we're going to look at on Wednesday, all the various things of cellular respiration, glycolysis, the transition reaction, the electron transport chain, the citric acid cycle before that. All of those reactions are the metabolic pathway. Okay? This ability to take something, break it down, and get stuff out of it. And by stuff, I mean energy, ATP. When we look at a metabolic reaction, we look at stuff on both sides. We have, I'm just going to keep it really simple. Okay, A plus B yields C. C plus D, okay? But a couple things on each side. The molecules that participate in the reaction are found on the left side here, okay? Those are referred to as reactants. A and B are reacting together to yield us C and D, okay? So these are my reactants, and these over here are my products, okay? They form, they participate in the reaction. They form as a result of the reaction, okay? So I may ask you, what are the reactants and products of photosynthesis? What are the reactants and products of cellular respiration? Obviously, I just ran through 4 different steps of cellular respiration, so you're going to have to know there's going to be different reactants and different products for each step of that cellular respiration pathway. Okay. I don't want to get too far ahead of myself, so I won't say that. Okay. So we have reactants and products. Right? Reactants generally on the left, products after that yield sign. This gives us energy. Okay. This metabolic pathway. Ultimately, you've heard the expression, right? You have to invest a little bit to make a little bit of money. That's what we have here in the metabolic pathway. There are certain steps within the metabolic pathway that require just a little bit of currency investment. So you have to put in a little bit of ATP. So you have to have a little bit in your coffers, in your reserve, to ultimately do the work, to ultimately get energy. Free energy is the amount of energy available to perform work. Okay? So to do all this stuff, we have to have a little bit of energy to kickstart the process. In cellular respiration, the first step, glycolysis, the first half of those reactions, you have to have an investment. Okay? I can't just take that glucose molecule and send it to the pathway. Okay? There's a toll. And that glucose, we have to pay the toll to kickstart the pathway. And if we don't have enough energy to start that off, pathway doesn't really go to fruition. Okay? So free energy is the amount of energy available to perform work. We store a little bit of free energy, guys. We store a little bit of free energy in our muscles. Okay? How much? Not a whole heck of a lot. I mean, we only get like 10 seconds worth. That doesn't sound like a whole hell of a lot, does it? But that little bit of 10-second amount of energy that we have stored in our muscles is enough that allows the other processes to kickstart and start handing over ATP to the skeletal muscles so it can continue to do work. Okay? Increase your rate and depth of breathing. Increase your oxygen so you can increase your cellular respiration, the amount of energy you're ultimately going to make. Getting your glucose out of storage or getting your fat out of storage from your fat stores. Okay? That little bit of time allows the other systems to get kickstarted and involved so it can give you the currency, ATP, to do stuff. Walk to your car after class, all the way across campus, something like that. When we look at reactions involving this process, there are two basic types of reactions here. Those that release energy and those that require energy. Now, if you just look at the first two letters of exergonic and endergonic, EX of exer, EX of exit. Oh, snap. Isn't that easy? Okay. Exergonic reactions release energy. These happen very easily. Okay. Our reactants have a lot of potential energy within them. And in this type of reaction, it's produced, released, we capture it, we utilize it. And then our products, C and D over here in this equation, that's water and CO2. We don't have a whole heck of a lot of anything within it. Okay, we've harnessed everything out of it, and those are my end results, my end products. Okay. Again, they just happen, these type of reactions. Okay, they occur spontaneously, so they just easily happen. Endergonic, ender, E-N, endo means within. Endo means within. Okay. So with endergonic, you need to put in energy. These don't just happen, okay? Whenever there's an input of energy required, there is a huge hurdle to get over for this chemical reaction to just occur. So these do not just happen willy-nilly. In reality, these two type of reactions are generally coupled together, okay? One is always happening in relationship to another. So an exergonic reaction releases energy. Endergonic reaction utilizes that stuff that's been just given off in an exergonic reaction to do its work, okay? And that's what they're trying to show you. I'll go back. And that's what they're trying to show you here, okay? Here's ATP, okay? It's going to be broken down and give off energy, okay? We break it down into adenosine diphosphate and an inorganic phosphate. And I have to put energy into the system to put that little P back on the other two P's. And that would be an endergonic reaction, okay? To create ATP. Break that bond again, release energy, and the cycle continues and continues. If I go back here, what the heck is ATP? I keep saying that term, adenosine triphosphate, okay? It's used to drive metabolic reactions. So all of this thing, and I have a nice little example here, okay? Beautiful lady here, holding a weight. There's the actin and myosin. This is right here in her skeletal muscle, here in her bicep, okay? There's Mr. ATP. It's ready to do stuff, okay? Goes up and attaches. Okay? The cascade of events have occurred, all right? Myosin docks up there with actin, and now she says, contract bicep. And there's our reaction right there. That power stroke move to help shorten the muscle, and boom, there you go. Little hills, okay? It's a coupled reaction. And again, ATP is the ultimate source of this currency. You can see what it's comprised of: adenine and ribose, which is just referred to as adenosine, and 3 phosphate groups, okay? And it's that last phosphate bond that gives off the energy. It's this bond right here, okay? Has a lot of potential energy in that one little bond there. So we break that bond and we release that energy, and that is the energy to do work. That is what allows actin and myosin to interact. That's what allows that skeletal muscle to shorten and contract, is the energy found in that little bond right there. We don't necessarily break this one at all, okay? That one stays there because there's a greater investment of energy to break that bond, okay? So we don't generally break that bond, all right? We just break that one. And this, again, like I said, it's a coupled reaction, okay? Where we break off that P and add it right back, okay? Here is a classic example down there at the bottom of a metabolic pathway. A to B to C to D, you know. I could have kept going through Z if we wanted to, but we don't want to do that. Okay? And these reactions, again, this has a lot of potential energy, and so it's just going to happen, okay? And we just go through all the intermediates to our ultimate end product, okay? Pathways, metabolic pathways, have to go in a specific sequence, okay? If I don't get C to form, I don't have D, E, and F. And obviously I don't have the end product, okay? So they have to occur in this sequence. If you take one step out of the metabolic pathway, the pathway folds, okay? And I don't get my end product, all right? And I have some examples, I think, at the end of this little bitty section here that looks at some of the poisons. This is how poisons work, where they interfere with the metabolic pathway to prevent end product production, okay? And that's what all of those words mean. I'm not going to go back through and read them all, but that's what we have here. Metabolic pathway occurs in a sequence, A to B to C to D to yada, yada, yada. Got to have C before D, okay? I can't go A, B, D. Can't do that, Okay, the pathway stops. Metabolic pathways can occur. Okay. They happen at a specific rate, but we have things inside of us that can help speed this up. Fabulous, awesome, because I want to make a lot of energy, because I want to be able to do my workout in the morning. Okay, and I need a good amount of energy to do that so I can make it through the whole thing. So we have these things called enzymes. And enzymes, we talked about that in the first unit. They are basically proteins, right? And they help to speed up chemical reactions. There it is. Okay. They are catalysts. Okay. And catalysts help to speed up chemical reactions. They bring the parties together faster. So this chemical pathway, this metabolic pathway, can happen faster. So these chemical reactions can happen faster, right? That's what enzymes do. That is what enzymes do. They're generally specific. They're generally unique, okay? These enzymes. This enzyme here, E1, can't function over here, okay? And to help get this there, usually all of these things are going to be present. Let me play this little video here. Oops. I need to hit a button. Sorry, guys. Hold on a second here. Okay. Okay, here we go. So let me try that again. And every time that interacts with an enzyme, it changes. Specific enzyme for a specific shape. That's why I have all of these specificities here with these enzymes. Because every time you can see, it helps to alter the shape a little bit of it. Okay, and we'll see that here in a second. Okay, we'll see this here in a second. All right, we are bringing together all of these. Okay, we are helping to form B because that enzyme helps to bring about change to get my end product, which is not A, it's B, it's something completely different. Okay, and now B is the new substrate for the second enzyme, and that alters its shape a little bit when it interacts with it and gives me a new product. Now that's the reactant. Interacts with the enzyme, gives me a new product, slightly different, okay? Slightly different. There we go. Where that substrate binds is called the active site. Where substrate binds. Okay. Active site of the enzyme is where the substrate binds. You saw in that little video, okay? That substrate always went to a specific location, okay? That specific location is the active site. Once it nestles in that specific shape, the active site, and it didn't show it very well, the active site changes just a little bit. And it's just that little bit of change that occurs there that actually, that's telling us that there's a chemical reaction occurring there. And just that little change in that active site, once that little substrate binds there, causes that substrate to now to change just a little bit. And now we get our new product. Okay. They call this the induced fit model. Okay. What does that mean? A slight alteration to achieve optimum fit to get my substrate. Okay. Okay. Here's my active site on my enzyme. Okay. Here's a substrate that comes in, and you can see it's just tiny smidge bigger, right? Just a tiny smidge bigger. So we just have a little bitty change, and that's that little bitty flex in is going to be enough to cause that substrate to undergo a chemical reaction, and now it is the new product. Okay. And now it is the new product. Enzymes can either be making new things, which is primarily what we've seen, okay? Or it can break down things. To make or to break. So in our example here, okay, this would be a synthesis type of reaction, okay? Two substrates. The enzyme brings them together, and we make our new product. This one, single, is broken into two. Okay. And we release those two molecules. Here's a picture. Okay. Here's the degradation. Here's my substance. Here's my substrate. Okay. Here's my little enzyme. There's the active site. Specific shape, specific shape. Form this enzyme substrate complex. And the enzyme is not destroyed. And now I have my two products, which looks exactly like that. They are just broken down now. Down here is the exact opposite. Okay. This would be a synthesis. Okay. Two substrates. There's my little enzyme, active site, specific shape, specific shape. It's going to say, oh, yep, fits. Again, enzyme is not destroyed in the process. It never is. And now there's my one product. Okay. Start with 1 and end up with 2. Start with 2, end up with 1. Okay. We are synthesizing a bigger product. We are producing 2 individual products, okay, from one thing. So 1 into 2, 2 into 1. Degradation and synthesis respectively. For these reactions, we have to have that potential investment sometimes. For reactions that just happen, okay, they generally have a very low investment, a very low energy of activation. Okay. Okay. And you'll see it probably shorthand on the exam as EA. Okay. And that's what we have here. Okay. This energy of activation, how much of an investment, how much ATP do I have to spend to get this chemical reaction to occur? That's energy of activation. Okay. If it doesn't take a lot of things, boom, happens easily, quickly, boom, gone. But it may need a slight input, and not every reaction that is exergonic is the same. We have some reactions in the body that happen very spontaneously, release a good chunk of energy. We harness that and do stuff. Some reactions take a small investment. Okay. Some pathways need a huge investment. Okay. So not every pathway is going to be the same. We talk about energy of activation. What hill do I have to overcome to end up with my product? How much energy do I need to put in to get my product? Enzymes are fantastic in this regard. They help to lower that energy of activation. They help to lower that cost. This makes our system a little bit more efficient. Again, it's not really that efficient, but it does make it a little bit better. Okay. Enzymes lower the energy of activation, so they help to lower that cost. All right. They help to lower that cost. And that's what they're trying to show you here. All right. This is energy of the reactant and energy of the product. Okay. So here, to release that energy, right, I have to have a huge investment. Okay. Energy of the reactant, energy of the product. To get that product, to get this reaction to occur, I have to have this much of investment. But if I add a little enzyme, okay, if I have a little enzyme present, you can see it helps to lower that hill that I have to overcome. It helps to lower the amount of investment that I have to put into this chemical pathway to get it to occur. Right? So I'm going to be able to harness more energy out of this thing. Where in the world do we get these things? Through our diet. Vitamins, minerals. They help us to create the enzymes that help us to make all this fabulous energy in cellular respiration and in photosynthesis, but I'm not a plant. Okay. So this energy of activation, how much of an investment do I have to have? Do I have to have a lot or a little? And when enzymes are present, they generally lower that investment cost. Okay. Some things we have a lot of, there are some pathways that we have a number of enzymes for, okay? And that helps us to harness a buttload of power, buttload of energy, all right? When our metabolic system starts to diminish, we don't make as much energy. We get tired. So what do? Well, we eat food, right? Eat something. Perks you up a little bit. Take 5-hour energy. What's 5-hour energy? Nothing but B vitamins. B vitamins themselves are not giving us energy, but what do they give us? All the little parts and pieces to make all these fabulous enzymes, which can now enter to the metabolic pathway and give you 5 hours of energy. Okay, make you feel awake and alert. And that's what they talk about here.
Remember that in a chemical reaction, the reactions interact to form a product. In this graph, we are examining the relationship between the amount of energy in a chemical reaction and the time it takes the reaction to form a product. As you are probably aware, chemical reactions are rarely spontaneous. This is because of an energy speed bump that occurs during the chemical reaction. This speed bump is called the energy of activation, and it represents an energy threshold that must be overcome before the product may be formed. There is a relationship between the energy of activation and the rate of the reaction. The greater the amount of activation energy, the slower the speed of the reaction. However, cellular processes often require that chemical reactions occur at an accelerated pace. To increase the speed of a chemical reaction, cells use catalysts called enzymes. Enzymes function by lowering the amount of activation energy that is needed in the reaction, thus reducing the level of the energy speed bump in the reaction. Since less energy is required, the reaction may occur at a faster rate. Notice that the enzyme does not influence the energy level of the reactants or the products, but only the amount of energy that is required during the process of the chemical reaction. Without the use of enzymes, many of our body's processes, such as digestion and the processing of nerve impulses, would simply occur too slowly. In other words, enzymes make life possible.
Enzymes make life possible. Love it. Okay, 100% correct. There are several factors that affect enzymes. And I think in one of your, one of those labs, you get into this. Okay, you get into this, maybe both of them, because they're both related to one another. Okay. But there are several factors, 3 big ones. Okay. How much substrate? If I increase my substrate, I can increase my reactions. Okay. There's more people there. Think of a room, and it's so much, this is the fun part. Okay. This is where I have you get up and we walk around the room. Okay. If we have 40 people in a classroom, there's going to be a lot of people kind of bumping in, excuse me, you know, trying to get around everybody. Okay. When there's fewer people in the room, I can walk around the room really easily and not bump into anybody. And when I do eventually bump into somebody, then I have a chemical reaction occur. So if I increase the number of substrates, that will increase the rate of collisions. And when 2 things come together and collide, they undergo a chemical reaction, right? So substrate concentration, if you increase the substrate, you increase the rate of enzymatic reactions. Temperature. Enzyme activity increases with temperature. Now, there's a fine line, and I believe I talked about this, you know, in the last chapter. Okay. The warmer the temperature is, the more collisions. They're speeding up and moving around. So they're going to collide faster. But if you increase the temperature too much, you destroy the shape of the protein. And when you destroy the shape of the protein, you render it useless. Okay? And you would destroy the enzyme. I wouldn't ask it, but Siamese cats, they have a mutation that cause enzymes to be active at cooler temperatures, and that's what's affecting their coloration. And that's what they're looking at here. Okay. Those that are able to stay warmer, they're going to be able to have an increased rate of reactions, which is good because it needs a lot of energy to run around and do stuff, keep its body temperature. pH is the other most important factor. All enzymes function within a certain range of pH. Okay. Pepsin functions best at a pH of 1 to 2. Okay. After 2, the effectiveness of pepsin in this case is an enzyme in your stomach. It doesn't start to, it starts to diminish in function. What does that mean? You don't break down proteins as effectively. Okay. Pepsin is only good in the stomach because of that very, very, very acidic environment. pH of like 1, 1.2, I think, is the stomach. Okay. Trypsin is secreted by the, and I'm not gonna, I don't expect you to know trypsin and pepsin. Trypsin is going to be secreted into the small intestine. It's not an acidic environment. It's alkaline, basic compared to the stomach. Okay. It only functions, as you can see there, at around, let's just say 8, optimally. You get below 7, you get above 9, too acidic, too alkaline. Trypsin doesn't work. You don't break down the proteins, which means you don't absorb the proteins. And now you have a deficiency of protein within the body. And that's going to affect growth, your rate of growth, okay, all that stuff. So pH, temperature, and the amount of substrate you have are the 3 major factors that really affect enzymatic speed. pH, again, it's not necessarily the pH of 1.2 doesn't necessarily speed it up, but it just maintains the shape of the enzyme. By maintaining that shape of the enzyme, it can function optimally. Too acidic, too alkaline. You alter its shape a little bit. It denatures. Okay. And when we denature a protein, that's when we destroy its function. Okay. If you cooked an egg this morning, if you kept it on the burner too long, there around the edge of the egg, you got that hard, brittly, it doesn't taste good. All right. That's a denatured protein. Okay. You've destroyed that albumin. Okay. And now you've destroyed that protein and it's useless. You're not going to eat it. It tastes like crap. It's not going to do anything for you anyway. Enzymes, they're great, right? Like the video said, enzymes are life. Okay. But enzymes will have things that they need. Okay. These guys, cofactors and coenzymes, are stuff that have to be incorporated into an enzyme for it to function optimally. And again, this is FAD and NAD. I'm going to highlight these 2 guys, okay, because those 2 are really important. Okay. You get that from niacin and you get that from riboflavin, the B vitamins. These 2 little cofactors help photosynthesis, help cellular respiration to make a lot of potential energy, a lot of ATP. If you are neglecting these vitamins, okay, these B vitamins, niacin and riboflavin, right? You are not going to be able to make enough energy quickly or effectively. Okay? So these are my vitamins and these are my minerals. Okay? These are the components that go into these enzymes to help the metabolic pathway. And if you come to nutrition class, I talk a little bit about that. We talk about all the vitamins and minerals, obviously, some more than others. And we talk about some of these factors that go into enzymes to help speed up the metabolic pathway to make a buttload of energy. There are hundreds of enzymes that vitamins and minerals help to make, and they help to speed up the metabolic pathway in so many different ways so we can get energy out of it, so we can do work. It's one thing just to sit here all day, but as soon as you're going to get up and move around and interact with the world, you need a lot more energy. And it's these cofactors and it's these coenzymes that help us to make these enzymes be as effective as they can. We can inhibit enzymes, which, is a good thing, okay? I don't necessarily need a chemical reaction to just continue forever, okay? So I do need these metabolic pathways to shut down, okay? Or slow down ultimately. And that's what we have here with enzyme inhibition. Enzyme inhibition is just a substance that inhibits the binding of an enzyme. And again, this is just to slow down the pathway, okay? I don't need metabolic pathways to continue at 1,000 miles per hour all the time. We have two ways that we can inhibit enzymes. And I added another picture here because it just, for whatever reason, the book didn't have one in there. So I added one on the next slide, I believe. So when we get there, that's what you'll see. Okay, I'll try to update it later today on Blackboard for you. We have competitive inhibition and we have non-competitive inhibition. Okay. With competitive inhibition, the substrate and the inhibitor both can, same shape, okay? So they can both compete, competitive, okay? The substrate and the inhibitor are both able to bind to its active site, okay? This is called competitive inhibition, all right? The inhibitor can be another enzyme, okay? Could be just any substance at all, okay? There are a number of receptors, obviously, all over the body. There are some receptors that only like one type of neurotransmitter. Right? But let's say you do marijuana or you do cocaine. Those drugs have chemicals within them that are the same shape as the specific substance that usually binds to a receptor site. So now you do that drug, it binds to that receptor site, and it is competing with the normal substrate. So now you get a cascade of events occurring because now that drug is binding to the active site, causing a chemical reaction to occur. Euphoria, whatever the case may be with the drug. Okay? So that's competitive inhibition right here. Okay? Substrate, inhibitor. Same shape, binds at it. Obviously, now the pathway can't go on. There's also this thing called non-competitive, all right? They don't have the same shape, okay? But the inhibitor in this case binds to another site, okay? And that's the allosteric site. Here's the allosteric site, okay? Here's the allosteric site. The inhibitor binds to that, and it causes a change in the active site. And now the substrate can't bind there. Not the same shape. Whoops. Okay, not the same shape. Okay, now it's all skinny, and this guy has a nice big fat active site. Okay. Change in shape at the allosteric site. Binding of that inhibitor to the allox site causes a change in shape of the active site. And now the pathway can't continue. And that's the picture here. This is the one from your book right here. Okay. There's the enzyme. There's the substrate. Okay. Binding with it. Okay. And that causes this to be active and blah, 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 blah. And then we have the metabolic pathway. But on this enzyme, you can see it does have another site, the allosteric site. Specific shape, specific shape. So in this case, the end product can bind to that allosteric site, inducing a change. And when it induces a change, you can see right there that now the substrate A can't bind. What happens to this metabolic pathway? It stops. So in this case, the end product is the inhibitor. Okay? And in a lot of our pathways, this is what we got. This is what we got, which is awesome, because now that shuts down the pathway. Again, I don't need the vast majority of any of these pathways continuing and continuing and continuing. Okay, I don't need it. There are a number of things that if we do interfere, it can be very deadly, and a lot of poisons function in that regard. And this is slide. What slide is this? 34. I'm not going to ask you anything off this slide for points. If I was to ask, it'd be for bonus. So don't kill yourself memorizing this stuff. I don't expect you to. Okay. But there are some substances that can interfere, like cyanide inhibits ATP production. Okay. Which will render muscles to not be engaged. Sarin functions at the neuromuscular junction. It inhibits enzymatic action, so it causes a different set of reactions depending upon the system. Warfarin inhibits an enzyme responsible for the blood clotting process. Warfarin's a drug. This is done for people that, for some reason, have blood clotting issues, and prevents those blood clots from forming. Okay. Usually they'll give this, during a surgery too, so the body's not fighting it when the surgeon cuts them open. Okay. So warfarin inhibits an enzyme responsible for the cascade of events of the blood clotting process. Again, destroy one little step in that cascade of events for the blood clotting process, and the blood doesn't clot. Okay. So not everything has to be doom and gloom, okay? Warfarin can be something to help keep people alive. This leads us into oxidation and reduction reactions. Now we are starting to get a little bit closer into what we're going to be looking at with photosynthesis and ultimately cellular respiration. And there we probably make it a little bit more of an emphasis in there. They're called redox reactions, but oxidation reduction reactions is what I'm going to try to just call it. Okay. We have these things called electrons, right? We saw these in the first unit. And we can pass them from one molecule to another. Okay, we saw that with covalent bonds, right? Ionic bonds. Sometimes they share unequally, sometimes one takes from another. Either way, okay? When electrons are passed from one molecule to another, we refer to them as either losing an electron or gaining an electron, okay? Are they oxidized? Do they lose an electron? Is it reduced? Gain an electron. Remember, electrons are negative, okay? So it gets more negative, gets reduced. For one to gain, one has to lose. So obviously these are both going to be occurring at the same time. There's a mnemonic to remember these. Students generally have a difficult time with this oxidation reduction concept, which is to say, I'm probably going to ask about it on the exam. So oil rig is the mnemonic that they've come up. Oxidation is lose. Reduction is gain. There you go. Pretty cool. Okay. And if you know that valence shell, then you can usually figure out which molecule is going to be the oxidizer and which one's going to be the reduced. Okay. I like to think of it this way as well. Oxidation means something's been broken down. Okay. So usually I think of it in terms of that regard. And when you lose something, you break down its shape a little bit. Okay. You've broken down its structure if it's lost. So sometimes I visualize that in my head. So here are these two processes. And this is ultimately what we've been talking about this whole time. The sun is the source. If I don't have the sun, then I don't have plants. No plants, no big animals, right? Plants harness that sun. Okay. They take our waste, CO2. And we don't really produce a copious amount of wastewater for them. But they get that from the ground, right? So they get the CO2 in the water, CO2 from us, water from the ground, solar energy from the sun, and they make stuff. They make glucose, carbohydrates. And as we'll see Wednesday, it's going to go to help make structures. It's going to be stored for later use. And it gives off a waste product, oxygen. Thank God, right? We take that oxygen. We can take that glucose. And we can use it now to make energy. So there at the bottom of that slide, you can see the reaction, the chemical reaction for cellular respiration. Glucose plus oxygen yields carbon dioxide plus water plus energy. It's the same thing. That's the same chemical reaction, it's just in reverse. CO2 plus H2O yields plus energy glucose and oxygen. And the reaction can go backwards as well. Okay. So how is photosynthesis and cellular respiration connected to one another? Starts with photosynthesis, right? We need the sun, our waste product, CO2, water from the ground. We can get glucose. We can get a waste of oxygen. And we utilize that oxygen. We utilize that glucose. And we can get energy out of it. And then we give off waste. CO2, exhale. And water. Sweat out, urinate, whatever. So these two things, although they're ultimately different, they are connected. We need the sun. We need plants. If we don't have plants, no one's capturing the sun. And no one's making energy out of it. No one's making carbohydrates for animals to eat. And then we eat the animals. Okay. And then we don't have enough energy to go out and run and to do stuff. So these two very different things, photosynthesis and cellular respiration, they're both dependent upon one another. Our waste product funds their action. Their waste product funds our action. Okay. So these vastly different things are connected. Sorry. And again, the carbohydrate made, glucose, goes to the mitochondria. Remember, plants have both. Plants have both photosynthetic chloroplast and mitochondria. Okay? Mitochondria are not something exclusive to animals. All right? Plants have both. Okay? So the plant is going to take that solar energy and make glucose. Glucose can be utilized for a multitude of things in a plant's structure, making energy. Because what happens at night? Sun goes down. How in the hell a plant's going to survive the night, depending upon the season, that length of time? From the glucose it's made throughout the day. Send it over to the mitochondria, and even though we don't have sun, plant can still make energy over the nighttime. Okay. And when we start looking at photosynthesis, we're going to see kind of this last concept right here. Okay. Energy is not necessarily cycling between these two little organelles. Okay. Number of steps happening within the chloroplast. And that product has a multitude of ways it can go. Some of it goes here, some of it goes here, some of it goes to the mitochondria. And then now it has to go through a number of steps. So we can harness the energy out of that glucose molecule and get us usable energy for the plant to do work and waste product that we can give off, and the cycle can continue. Some good potential short answer questions here. Okay. The laws of thermodynamics would be one. Okay. Another thing could be factors affecting enzymes, substrate concentration, temperature, pH, cofactors and coenzymes, if you want to talk about those. Another good short answer question. Maybe another one would be, comparing comparing the difference between competitive and non-competitive inhibition. Maybe. But if you could answer that as a short answer question, then you could answer any way I would ask it. Multiple choice, true, false, matching, fill in the blank. You can answer any question about it, obviously, if you can write it all out. Okay. So this is our first foray into what is ultimately going to be talked about on Wednesday and Monday next week, the making of ATP. We're going to put together all of these fun little things that we looked at in the first unit with the little organelles. We'll look at what we talked about in chapter 5 with the plasma membrane, because we're going to talk about some carriers that are found in the plasma membrane. And that's how we are going to be able to create a good amount of ATP potentially in certain situations. We'll be talking about exergonic and endergonic reactions ultimately without saying those words with photosynthesis and especially cellular respiration. Okay. So in the next 2 chapters here, we're going to be putting into application a lot of this stuff that we've already looked at. We may not be specifically calling it by those words, okay? But now we're applying what we know about our organelles, and we're applying what we know about the proteins embedded within the plasma membrane. And we're going to be applying what we know about metabolism and reactions that give off energy and reactions that require an input of energy, okay? So in these next 2 chapters, we're going to be applying all this stuff that we've been talking about, discussing. You've been doing labs on it all, okay? So now we're going to be putting this stuff into action, all right? We're going to be putting this stuff into action. So that photosynthesis, especially cellular respiration, really fun, okay? Really fun. In nutrition class, we expound upon this just a little bit. When we look at the macromolecules and the vitamins and the minerals, those micronutrients that are involved in metabolism, and how food, how carbohydrates enter into the metabolic pathway, how proteins enter into the metabolic pathway, how fats can enter into the metabolic pathway, and how that metabolic pathway can be enhanced by vitamins, minerals not being there. Okay. So in nutrition class, we take it into a different foray, different step, and it's really fun. A lot of application there. And then for anatomy and phys, we'll go slightly a different way with it. We talk about how the body breaks it down. Well, I do that in nutrition too, but I get a little bit more in depth into how we break down these nutrients to enter into the metabolic pathway, to fund skeletal muscle activity, to be able to do stuff, to fund nerve impulse conduction. So this is the basic building block for what everything is in science, is this little unit. Because without power, without ATP, I can't do crap.